Hello students, today we will be touring an animal cell. If you are here to see cell walls, chloroplasts, and large vacuoles, you are in the wrong place. That is the plant cell tour, which is 3 doors in the opposite direction. Now then, those of you who are here for the animal cell tour, we will visit the cell membrane first. The cell membrane consists of a bilayer of phospholipids. The hydrophilic ends of the bilayer face the inside and outside of the cell. The hydrophobic ends face away from the inside and outside of the cell. There are several embedded proteins within the cell, which help transport large charged particles through the membrane. Smaller particles can pass through the membrane during diffusion or osmosis with ease. We are now moving away from the cell membrane and towards the Golgi apparatus. The Golgi apparatus is a stack of membrane-bound organelles that prepare various things within the cell for transport throughout the cell via vesicles. We are now coming up on one of the mitochondria. The mitochondria are membrane-bound organelles that provide the energy a cell requires to move. Moving on from the mitochondria, you will see a centriole. Centrioles produce the microtubules that make up the cytoskeleton of the cell. Centrioles are not found in plant cells. As we leave the centriole, we will pass over some unbound ribosomes. Ribosomes are found in eukaryotic cells and are tiny round organelles that generate proteins from RNA. Ribosomes are composed of protein and ribosomal RNA. We are now coming up on the smooth endoplasmic reticulum. The smooth endoplasmic reticulum has a wide range of functions, which include carbohydrate and lipid synthesis, and it also serves as a transitional area for vesicles, and it produces enzymes that help detoxify certain materials. We are now coming up on another organelle that is unique to animal cells. It is the lysosome. Lysosomes are necessary for intercellular digestion. Leaving the lysosomes, we are now coming up on the rough endoplasmic reticulum. The rough endoplasmic reticulum is called rough because it is covered with millions of ribosomes. The ribosomes make it appear bumpy under a microscope. The rough ER both creates and folds protein. The large spherical thing up ahead is the nucleus. We are going to go inside the nucleus to get a better look at what it does and what is inside it. The nuclear envelope is a highly regulated membrane barrier that separates the nucleus from the cytoplasm in eukaryotic cells. The nuclear envelope has pores that regulate what goes in and out of the nucleus. In the nucleus we find chromatin, which is a complex continuum of DNA and proteins packaged together to form genetic material of chromosomes. The nucleus also contains the nucleosis, which has proteins and RNA that are not related to the ribosomes. We are now going to leave the nucleus. As we leave the nucleus, we are going to check out this fluid that suspends all of the organelles in the cell. It is called cytoplasm. It also acts as a buffer and transports genetic material throughout the cell. As we exit the cell, we are going to stop and look at the cilia. The cilia are hair-like organelles that branch out from the surface of the cell and help with movement. Thank you for choosing this particular tour group, which shall remain nameless for legal reasons. If you enjoyed your tour, it was to Quinn Thomas's credit. If you disliked the tour, it was all Nuri's fault. I hope you have a wonderful day.